The aim of my project is behavior checking of complex synthetic and natural biological systems. In systems biology, computational models are constructed from observations of biological systems and are used to make predictions faster and cheaper than experiments performed in a wet lab. In synthetic biology, researchers start from a computational model which has a desired behavior and use it as a design for engineering a useful synthetic biological system. In both cases, models need to be validated before using them in practice. Traditional model validation approaches capture the evolution over time of numeric properties such as concentrations. They are appropriate for small-scale systems such as intracellular pathways or simple cellular mechanisms. For solving complex problems, we need to scale up and design models beyond the single cell, which cover multiple scales. My PhD project aims to develop validation techniques for such multi-scale models. The novelty of the approach is that it captures how spatial structures and patterns evolve over time, which is not covered by traditional approaches. The contributions of my project are a formal logic for expressing spatial-temporal properties, a standard for representing spatial-temporal behaviors or time series data, visualization and analysis tools, and a software called Behavior Checker, which verifies if spatial-temporal properties are valid considering a given set of behaviors. The Behavior Checker was designed to use behaviors generated from model simulations, but also from wet lab experiments. The benefits to industry are two synthetic biology applications. The first application is increasing the yield of solid-state fermentation processes using microbial populations. This is achieved by constructing a model of the fermentation process and refining the parameters until a target yield is reached. Colonies grown inside fermenters have a spatial structure which needs to be captured by the model and validated using the behavior checker. Such models could enable replacing batch with continuous production. This may reduce total cost by 15 to 50%. The end goal of this approach is efficient production of biofuels. The second application is the production of novel low-volume, high-value products using collaborating or competing populations of bacteria. Interacting bacterial populations have a spatial structure which can be optimized using models and validated by the behavior checker. This approach could be used for the production of novel antibiotics which meet the needs of treating antibiotic-resistant bacterial infections. In conclusion, behavior checking could enable the construction of complex, valid models. Using such approaches, synthetic biology will be able to heal us, feed us, and fuel us.